The foundation of human civilization rests on the shoulders of science and technology. Back in 1944, this realization impelled the Nizam of Hyderabad to issue a farman directing his government to set up Central Laboratories for Scientific and Industrial Research (CLSIR) at Hyderabad, functioning from the campus of Osmania University. After the independence, political and scientific leadership of the country resolved to integrate CLSIR with Council of Scientific and Industrial Research, popularly known as CSIR. The institute continued to majorly focus on industrial development, agricultural needs, and drug discovery. The freedom movement had seen a dream to build a modern, secular, technologically advanced, and scientifically tempered India, and the institute committed was to this objective.
This organization has roots way back to 1944, where the erstwhile Nizam has issued a farman that this region should have a chemical laboratory to cater to the needs of the region. We are very proud that post-independence, CSIR has taken over our lab. This lab was renamed as Regional Research Laboratory Hyderabad. This is one of the regional laboratories of CSIR. As soon as this laboratory was formulated, the mandate was set by the then Government of India that it should, while catering to the needs of the chemical community, also should cater to the needs which are risen to the country. Incidentally, when we got independence, we had starvation, and then we need to bring new pesticides to this country, and those pesticides have to cater to the needs of farmers at the cost what they can afford. I am very proud to state that our organization has made pesticide industry flourish in the early 50s and 60s, and of course in the early 70s also, wherein over 60% of the Indian pesticide industry was using the technologies developed by this great organization.
During the past 70 years, the Institute has expanded its activities in many areas. Today, it is internationally recognized for its contributions to chemistry research and is an ideal place for germinating ideas and taking them to commercial stage through the state-of-the-art research and development. With changing times, the Institute has always kept up by realigning its research focus on areas of national and international importance. The Institute's research and development activities could be categorized as strategic research, private goods, societal goods, and public goods. It is important for any nation to become science and technology sufficient in order to develop. When India achieved its independence, the focus of R&D was to uplift the country's socio-economic position by upswinging the agricultural produce. But in no time, we understood the importance of producing our own technologies and securing our position in a globalized world. Developing scientific temper and applying it on areas of strategic importance to the country was the need of the hour, and different divisions of CSIR IICT responded to it. Various projects were developed to suit the requirements. Equipped with 300 MHz to 700 MHz spectrometer, CSIR IICT has the largest nuclear magnetic resonance NMR base in the country. It makes CSIR IICT a preferred destination for industry and academic institutes for studying molecular design and transient state chemical reactions. The division was approached by DRDO to study complex materials which are used for constructing large objects. Recently, a portable NMR device was conceived, designed and invented for on-site application that helps in investigating depth profile of objects. CSIR IICT has conducted research and development on polymers for various end users. It extends services to industrial clients and develop processes for paints, coatings and allied fields. The division has been a pioneer in developing and transferring technologies. When strategic sectors of India needed high-performance materials for self-reliance, processes and technologies for such materials were developed by the Polymer and Functional Materials Division. They developed a much-required resin and provided to Aeronautical Development Agency, who needed low-loss materials for fabricating radomes for light combat aircraft. Similarly, canister epoxy resins for BrahMos missiles was also developed by CSIR IICT and transferred to DRDO. They also provided suitable fluoroelastomers for Terminal Ballistic Research Laboratory, Chandigarh, for making elastomers with exceptional weatherability for plastic bonded explosives. Several other processes and products like hydrophobic rain erosion resistant coatings, polyacrylonitrile resin for making carbon fiber were also developed as per need of strategic sector. India is dependent on advanced countries for its aviation lubricant requirement. Hence, CSIR IICT has launched a project for development of technologies for synthetic aviation lubricants from indigenous resources. Very recently, Lipids Group, in collaboration with several other groups of CSIR IICT, has successfully completed the process development for 2 such candidates. Analytical facility comprising of highly sophisticated equipments has been created for lubricant testing, and a pilot plant for the preparation of biolubricants has been established. Development of lubricants based on several non-edible oils like Jatropha, Karanja, Castor, Sal, etc. for different applications is also being actively pursued, and several patents have been filed in these areas. The division has been working relentlessly to provide strategic support to the country. Development of technology and its successful transfer to indigenous industries has been a vital responsibility that CSIR has taken up. CSIR IICT Hyderabad has developed many technologies as per the requirements of Indian industry and has also attracted global partners to its innovative and benchmarked technological exploits. The process development of azithromycin AZT, one of the most expensive drugs for the treatment of AIDS, was developed indigenously for the first time, bringing down the costs in the world market. A new and highly cost-effective route for beta-azithromycin, a crucial intermediate for AZT, was also developed from D-xylose. The technology has been successfully transferred to private players, and other bent-scale processes were also developed for norfloxacin, ciprofloxacin, naltrexone, and others for several clients both from India and overseas. Hyderabad has been identified as a pharma hub based on the contributions of CSIR IICT in supporting the pharmaceutical industry. To help find new scaffolds for the treatment of diseases affecting human race, the Institute has established National Mall Bank, a unique facility for storing the samples and sharing them for screening. The focus at present is on finding cures for tuberculosis, cancer, and Alzheimer's or Parkinson's disease. They are also involved in the development of misoprostol drug, which is used for family control and other uterus-related problems. Due to acute shortage of edible vegetable oils in India, the focus shifted on development of processes for non-conventional oils like rice bran oil. India is a major producer of rice. Research activities were directed towards development of edible grade oil from rice bran. The processing of rice bran oil is associated with many technological challenges. Lipids Group actively pursued R&D on this unique, nutritious oil and made remarkable achievement by developing technology for enzymatic degumming of rice bran oil with the funding of Technology Mission on Oilseeds, Pulses and Maize, and the technology was transferred to 19 industrial houses and 8 project engineering companies, which made a deep impact on the vegetable oil industry in the entire country. Immediately after independence, the country faced acute shortage of food grains. CSIR IICT started working in the area of pest management and developed cost-effective and environmentally friendly processes for agrochemicals. The institute has also worked in developing technologies as alternate options to minimize use of pesticides. These technologies are being provided to farmers to improve crop yields. Diazinon, Butachlor, Chemaxin, BHC are a few pesticides developed by the division. They are working relentlessly on clean and green processes to use biopesticides as a substitute. Any technology until used for the good of the masses is not considered a noble technology. It is the vision of CSIR IICT to develop and transfer technologies for the greater good of the public. They have worked relentlessly on projects ranging from public health, agricultural technologies, environmental degradation, and urban lifescapes. The motive has always been progress and upliftment of human lives. Contributing immensely to rural welfare through water purification, CSIR IICT has developed projects. It is also actively involved in spreading of technological awareness and laboratory development. They have commissioned several membrane pilot plants of varying capacities based on electrodialysis, nanofiltration, gas permeation to facilitate solvent recovery, effluent treatment, and gas purification in pharmaceutical, textile, and petrochemical industries. Design and installation of 10 model defluoridation plants of 600 to 4,000 LPH capacity for purification of groundwater for fluorosis-affected villages of Andhra Pradesh, Telangana, and Tamil Nadu was also undertaken by the Institute. For the benefit of the rural populace, useful information on the topics of agriculture, health, education, pest disease information system, and various other aspects is audio and video programs called as Samadhan Kendra, catering the needs of huge farming community in India. NVIS Center on Bioinformatics Vector Control, sponsored by the Ministry of Environment and Forest, was set up by CSIR IICT. The Chemical Biology Division works for the primary screening of small molecules against cancer, CNS, diabetes, and cardiovascular diseases. The Liquid Handling System, or commonly known as LHS, is state-of-the-art equipment housed in this division, which is used for handling large numbers of compounds. The Mall Bank facility harbors various natural and synthetic molecules prepared at CSIR IICT and elsewhere. The facility is the first of its kind in the country, which integrates synthetic organic chemistry, structural biology, nanotechnology, biochemistry, and cell biology under one roof. It currently serves the needs of Medicinal Chemistry. The Medicinal Chemistry and Pharmacology Division at CSIR IICT aims to actively engage in developing affordable therapeutics for human ailments, particularly relevant for Indian scenario. They are credited to innovate green and economically viable drugs such as mefloquine, sulbactam, carbidopa, dilatazine, ketotifen, stevioside. The division is actively involved in silico drug design, green chemical synthesis, biological testing, microbiology, pharmacokinetics, pharmacodynamics, animal model studies, and toxicity evaluations, and is working parallelly with national and international pharmaceutical companies. An innovative high-rate biomethanation digester called Self-Mixed Anaerobic Digester, SMAD, was developed at CSIR IICT for the treatment of organic solid waste. It was demonstrated at Livestock Research Station, LRS. A modular digester is designed based on scientific doctrine of environmental biotechnology coupled with advanced engineering principles to accomplish superior process efficiency for safe discharge of garbage. This technology is field tested, and currently these digesters are working at housing colonies. For helping the farmers in the country, the stress of research changed its due course towards developing new pesticides without producing any side effects. It was realized that semiochemical, a naturally occurring volatile organic chemical released by plants, insects, and other organisms, helps in specific communication purpose. They include pheromones, intraspecific activity, and allelochemicals, interspecific activity. Pheromones are chemical substances, usually mixtures of chemicals, that act as messengers of communication between individuals of the same species, eliciting behavioral responses. Our species pheromones in integrated pest management (IPM) practices has been recognized as an important and promising tool in providing ecologically safe and environmentally friendly control strategy. Particularly pheromones are found highly suitable in the management of miner, borer, and dweller category of pests. Focused on the development of cleaner processes for utilization of low-grade Indian coal, Coal and Gas Unit at CSIR IICT carried out a number of research projects on low-temperature carbonization of coal, coal tar distillation, briquetting of coal char, moving bed pressure gasification of coal for power generation, hot gas cleanup for dust removal, desulfurization, denitrification, and dealkalization of coal-derived fuel gas. In the backdrop of the oil crunch created by the Arab oil embargo, the division took up the coal gasification project and successfully erected a pilot plant. To overcome the hurdle of corporatization of technology, Fluoroorganic Division was established at CSIR IICT in the 90s. Chlorofluorocarbons, CFCs, were banned world over due to ozone depletion, and thus hydrofluorocarbons, HFCs, were identified as substitutes. But the technology of developing them rested with the first world, making it a prerogative on the developing world to harness an indigenous technology. The Fluoroorganic Division at CSIR IICT worked on chemical like 1112 tetrafluoroethane, HFC 134A, for developing suitable technologies. The developed technology for HFC 134A from lab scale to semi-commercial scale was transferred to Indian companies, which were subsequently commercialized. Disease modeling of vector-borne diseases like filariasis, malaria, Japanese encephalitis, and dengue is developed by CSIR IICT. It is pioneering work in the area of public health. Biological databases on mosquito and mosquito-borne diseases was developed and transferred to Health Department, Government of Arunachal Pradesh and Andhra Pradesh. Prioritization of disease endemic zones has been done through self-organizing maps. This technology was developed and transferred to Ministry of Health and Family Welfare, Government of India. The institute has developed an integrated disease forecasting system for the control of malaria in Mizoram and Sikkim. Anopheles and Culex mosquito identification through expert system application was developed and transferred to DRDO. A GIS-based application was developed for identifying disease hotspot regions in endemic zones of India. Besides these, CSIR IICT has several divisions, which provide services and facilities to various departments. The versatile pilot plants erected at the CSIR IICT campus are a witness to many technologies that have been successfully upscaled from lab to industry level. Without these plants, commercialization of various processes developed by CSIR IICT would not have been possible. Upscaling is an important link between lab and industry, which inculcate confidence among both the scientists and industry leaders. Working silently but ferociously are certain departments which add to the knowledge base and act as facilities for the Herculean work undertaken by CSIR IICT. Center for Molecular Modeling demonstrated the cation P as the strongest of non-covalent interaction and probes its variation as a function of size, nature of ion, solvent, and environment. They work around the prediction of relationship between material structure and performance. It provided insights into the nature of catalyst and help drug design. The Design and Engineering Division is like a skeletal system for transferring technologies developed at CSIR IICT. The design team consists of process developers, process designers, and detail designers. They work together from the laboratory scale through bench scale, pilot scale, and finally the commercial scale. As the team is closely associated with the process, they have an insight to intricate details based on which they help in both horizontal and vertical technology transfer. The Analytical Chemistry Division provides qualitative and quantitative analysis services, industrial and academic bases. The work undertaken by them is usually separated under mass spectrometry and separation sciences. They have worked relentlessly, worked on differentiating chiral isomers, mass spectral studies on unnatural amino acid peptides, non-covalent interactions between biomolecule, stress stability studies, and pharmacokinetics of drugs and pharmaceuticals. Nanomaterials Laboratory is a part of the Inorganic and Physical Chemistry Division and has undertaken development of material and devices. The expertise lies in material synthesis, material characterization, device fabrication, and material designing. Currently, the laboratory is engaged in 3 projects: formation of molecules to material device with specific sensors for carbon dioxide detection, nanomaterials for safety which uses photodynamic therapy during cancer cells, and intelligent coatings for antibacterial and anti-corrosive activities. Nanomaterial Laboratory has collaborated both with national and international agencies for developing nanomaterials and processes. X-ray crystallography, a facility for serving all CSIR IICT divisions, is known for its R&D activities on drug polymorphism such as sulfonamide, sertraline HCl, palonoxetine, aripiprazole, tamsulosin. Its core competency lies in chemical crystallography and material characterization using powder X-ray diffraction of fluorescence crystal structure analysis on compounds of biological relevance and pharmaceutical importance. Engineering Services Division is mandated to provide operation, supply and maintenance of utilities. It also manages a large central workshop comprising of a machine shop, metal joining and sheet metal fabrication unit, a captive foundry and forge facility, pattern making and carpentry, millwright shop to provide vital engineering support and mechanical and electrical maintenance to R&D activities of the Institute. The division has been a part and parcel for many ongoing projects. During the past more than 70 years, scientific, technical and administrative community working at Indian Institute of Chemical Technology has faced many scientific and technological challenges and have cracked complex problems. It has contributed to international pool of scientific knowledge.
Today, science has no boundaries. To make sure that we are on the global platform, CSIR IICT has always collaborated with best universities in the world. We collaborate with Mayo Clinic in the US on the oncology programs. We collaborate with RMIT, Royal Melbourne Institute of Technology, a university in Australia, to have joint PhD programs. We collaborate with University of Rennes in France, CNRS. We have a joint laboratory on the campus called Indo-French Joint Laboratory for Sustainable Chemistry. This joint collaboration has resulted in over visit of 200 students from France to CSIR IICT. They got the exposure of science, what we do. They also got the cultural exposure. Today we build networking programs. Today I am proud to state that this joint program has resulted in identifying some of the natural products which show activity in Alzheimer's disease, and I am very confident in the next 4 or 5 years this program would become a role model program of the international collaborations.
The scientists and technologists working at CSIR IICT even today stand like a tight fist to face the challenges to tomorrow and are busy building our scientific temper and an advanced country.